The overall goal of this assay is to enrich targeted transcripts for next-generation sequencing. This targeted RNA sequencing assay is a multi-step procedure that begins with the depletion of ribosomal RNA from total RNA, followed by chemical fragmentation and synthesis of complementary DNA, preparation of barcoded libraries, hybridization and capture of targeted transcripts, multiplex sequencing, and ending with data analysis. This video will demonstrate the procedures for hybridization, target capture, and post-capture PCR amplification for 4 samples.
So this method can help answer key questions in molecular biology, such as identifying gene fusions, single nucleotide variations, relative gene expression, and alternative splicing involving targeted transcripts. The main advantage of this technique is that it is customizable for any genes of interest.
To begin the procedure for hybridization, remove from the -20°C freezer and thaw on ice the following: custom hydrated probes, COT1 DNA, universal blocking oligos, and adapter-specific blocking oligos. In a low-binding 1.5 milliliter tube, combine 500 nanograms of DNA samples, 5 microliters of COT1 DNA, 1 microliter of universal blocking oligos, 0.5 microliters of 6 nucleotide adapter-specific blocking oligos, and 0.5 microliters of 8 nucleotide adapter-specific blocking oligos. Place the sample tube in a vacuum concentrator with the cap